Good morning, children. Today we'll start the second chapter, Structure of Chromosomes, Cell Cycle and Cell Division of Biology for Class 10 students. Here we will be discussing about the types of cell division, that is mitosis and meiosis, structure of chromosome, need of cell division and so on. So far you have learned the structure of a cell, isn't it? The structure of a cell is composed of component of cell wall, cell membrane, cytoplasm, cell organelles, etc. So one of the organelles is of nucleus, and inside the nucleus we can see the genetic material. You had learned in standard 9 some aspect of that. So now let us understand first of all the discovery of chromosome and the structure of chromosome. Who discovered chromosome? Chromosomes were first discovered by a German scientist known as Walter Flemming in 1882 when he was observing an amphibian known as salamander, the larval stage of amphibian, that is salamander larval stage. There he could observe some of the thread-like structures appear to be repeatedly dividing. And he coined the term for that mitosis. Mitosis literally means thread. So division is the thread like structures are dividing, and coined the term mitosis. So chromosomes were discovered by Walter Flemming in 1882 in the salamander larvae. Then structure of chromosome. When you observe a normal undividing cell, living cell under the light microscope, nothing would be visible in the nucleus, whereas the same cell, when you stain and observe under the microscope, you would be able to see some thread-like structures in the nucleus, and these thread-like structures are known as chromatin fiber. This chromatin fiber you can see when a cell is in the interphase. This all we will be learning in the next session, cell division, what is it and all. So when the same cell enters into the cell division, that is the prior or the first stage of cell division, these chromatin fibers condense to form thickened structures that is known as chromosomes. So chromosomes are visible only during the time of cell division. That is the first stages of cell division we can see chromosomes. Then what is a chromosome composed of or made up of? Chromosome consists of the structure, only the structure. Here two arm-like structures we can see. These two arm-like structures are known as chromatids, and these two chromatids are attached by means of a point. A constructive part is there. This point of attachment is known as centromere. So one of the functions of centromere is of attachment of chromatids. Another one function is there for the centromere, that is during the time of cell division, spindle fibers are also attached on the centromere. So what is this actually? The chromatin fiber or chromosome composed of. So chromosomes are actually what? Chromosomes are highly condensed coiled chromatin fibers. So highly condensed coiled chromatin fibers form the chromosomes. So what is this chromatin fiber made up of? It is composed of two substances, namely DNA and histone. It is a protein molecule. So their arrangement, what is their arrangement? Here you can see the DNA strand is coiling round or winding around the histone molecule. 8 molecules of histone are wounded by means of a DNA strand, and this one form a particular structure known as nucleosome. So nucleosome is of the coiled DNA strand winding the histone molecule to form nucleosome. So in human being, one single chromosome contains near about 1 million nucleosomes. So you have to imagine or think that this nature or the size of the cells are of microscopic. So inside this microscopic, again the cell, small nucleus. Inside the nucleus we have these chromosomes. Human being, we have 46 chromosomes in a cell. In a non-reproductive cell, we have the 46 number of chromosomes. So this is the arrangement. Here you can see this is the DNA strand. So now let us understand the structure of DNA strand. DNA is a single large molecule, and so it is coming under, it is known as a macromolecule. And their structure or model was studied by many scientists, and the most widely accepted model is of Watson and Crick's model. It is known as helical model. So here a DNA molecule is composed of two strands, or it is a double stranded molecule, wounded or spirally coiled, like looking like a ladder-like structure. This structure is known as, or it is discovered by Watson and Crick. So it is known as Watson and Crick model. And here you can see spirally coiling, one coil. This one is a complete coil from here to here. This is a single coil, one turn or complete turn. And what is it composed of? Each single strand, this is one strand and this is another one, that is double stranded. So each single strand is composed of three components: a phosphate molecule, a pentose sugar lengthwise, and as a rung or as a ladder, a nitrogenous base. This is what a DNA strand is composed of. Okay, and these three components, a phosphate molecule, pentose sugar and nitrogenous base, together form a nucleotide. So DNA strand is actually repeated chains of these nucleotides. So here you can see one nucleotide, another one, two nucleotides are there, composed of a phosphate molecules, a sugar, that is a pentose sugar, and a nitrogenous base. So this is the one strand. Opposite to this, complementary to this, another one strand is also here. So that is the double stranded structure. Okay, and here the nitrogenous bases of DNA. DNA is deoxyribonucleic acid. You already know the full form and all. So DNA is composed, the nitrogenous bases of DNA are of adenine, guanine, cytosine and thymine. And this pairing is always adenine pair with thymine by means of hydrogen bond, that is of 2 hydrogen bond. Then guanine pairs with cytosine by means of 3 hydrogen bond, that is here 2 line you can see 3. Any of the models of DNA, when if you observe, you can see AT and CG, AT by 2 bonding, hydrogen bonding, and CG by 3 hydrogen bonding. Vice versa also, AT also, TA also, CG and GC, like that it is arranged. And here chromosomes, this is a part of chromosome DNA, and here we have these all are the sequences, number of sequences we can see. This nucleotide sequences form the genes. So what are genes actually? They are the carriers of hereditary character, units of hereditary carriers. So specific sequence of nucleotide form the genes that encode a particular protein. Okay, these all structures in detailed way and all you will be learning in higher classes. Now, prior to the cell division, this DNA should replicate or divide. Then only the newly formed cell will be having the DNA molecule. So for this, the duplication or the replication of DNA takes place. How is it? It is given in your textbook. Let us see. So during the interphase of cell cycle or prior to the cell division, DNA molecule duplicate. It is for their equal distribution of the DNA molecule in the newly formed cell. And for this replication, first of all, the double helix or the coiled structure opens. Here you can see the opening or uncoiling of the strand. So these two strands are opened here. Here one strand, here these are the two old strands. And complement to this, another one new strand is formed. Here also another one new strand is formed. So new strands are formed complement to each other. Here thus we get a newly formed DNA. So each new DNA contains one original strand or parental strand we say, and another one new strand. Here also one original strand and one new strand of DNA. Here one original strand and another one newly formed. This replication also it is a highly complex process, and you will be studying these all things in the your higher classes. Okay. So the next portion of this one we will be continuing with the next video, cell division types, need of cell division and so on. Okay. Thank you.